Hi, I'm Elaine Vickers and welcome to my video series on cancer treatments. In this video, I'm going to explain why antibodies are used as cancer treatments. I'll start by briefly describing what antibodies are and then explain how we can harness some of their amazing properties to create effective cancer treatments. Antibodies are large Y-shaped proteins. They're made by a type of white blood cell of our immune system called a B cell or a B lymphocyte. B cells produce antibodies to help fight off infections, and that antibody usually sticks to something on a virus or a bacteria, something harmful that our immune system wants to destroy. Each antibody protein has two identical tips that stick to a specific target. Once the antibody attaches to its target, the back end of that antibody acts like a little signal, attracting other parts of the immune system, like white blood cells, which then attack and destroy whatever that antibody has become attached to. When we use antibodies as cancer treatments, they are almost always monoclonal antibodies. That means every copy of the antibody in the treatment is identical. These identical antibodies are made by one clone, a monoclone, of living cells. These cells are essentially a factory, producing billions of copies of the antibody that we want to use as our cancer treatment. The final product does not contain the cells that produce the antibody, but because living cells were used in the manufacturing process, we call it a biological therapy. Antibody treatments for cancer are very similar to the natural antibodies our immune system makes, but with one crucial difference. We have chosen the target. Scientists pick a suitable target by searching the surface of cancer cells for proteins that cancer cells need to survive, but that healthy cells either don't make or they make it, but they don't rely on it. The goal is to design an antibody that attaches to its target and directly kills the person's cancer cells. But what if the target is there on the surface of cancer cells, but it's not necessary for their survival? Well, we can still use it as the target for an antibody-based treatment, but we will need to find a way to increase its ability to kill cancer cells. One way of doing that is to create an antibody-drug conjugate. This is when scientists have chemically attached multiple copies of a chemotherapy to an antibody. The aim is that the antibody delivers chemotherapy straight to the person's cancer cells. There are other ways of using antibodies too. Sometimes we rely on the natural ability of antibodies to recruit in white blood cells of our immune system. This works best against blood cancers like leukemia or lymphoma or myeloma, where helpful white blood cells like macrophages and natural killer cells are often present and can be recruited by an antibody-based treatment. By the way, if you do like my teaching style and want some education for your team, then please do get in touch, and there are links in the description below. This approach of using antibodies to attract in and activate white blood cells doesn't work as well against solid tumours, because solid tumours often contain white blood cells that are protecting the cancer rather than wanting to fight it. Recently, scientists have started re-engineering antibodies, modifying their structure in clever ways. One exciting result of this is a group of treatments called bispecific antibodies. These can attach to two different targets. For example, one part of the antibody might bind to a cancer cell and the other to a T cell. They can take various physical forms. They might retain the Y shape of a normal antibody, like the one shown here, or they can be much smaller, just containing the tips of two different antibodies, like this. The goal is that the bispecific antibody connects together cancer cells with T cells of the immune system, and the T cell then kills the cancer cell. These treatments are sometimes called T cell engagers. We can also use antibodies to help T cells attack cancer in a different way through treatments called immune checkpoint inhibitors. there's a separate video about this group of treatments. You might be wondering, are antibody treatments targeted therapies or are they immunotherapies? The answer is they can be either. In fact, a few of them are both. An antibody might directly kill cancer cells and have ability to recruit in white blood cells too, making them a blend of a targeted therapy and an immunotherapy. It all depends on the antibody's target and on the way it has been designed. To recap, antibodies are Y-shaped proteins made naturally by our immune system to fight infection. We can use antibodies as cancer treatments by designing them to attach to a target of our choosing. Sometimes we're using an antibody-based treatment to kill cancer cells outright, but we can also use them to create a cancer-fighting response from the immune system. Depending on the antibody's target and its design, the treatment might be classed as a targeted therapy or as an immunotherapy, or both. Don't forget, if you like my teaching style and you want some education for your team, do find me on LinkedIn or contact me through my website. There are links in the description below.